All right, I would like to share some information with you about blood. Um, this is our 16th unit in physiology, and I think for the most part, you guys probably have heard most of this stuff before, but because we're doing talking about the circulatory system, pardon me, um, and talking so much about moving blood around, I want to make sure that we have an understanding of the components of blood. So, as I'm sure you recall from anatomy, blood is the only fluid tissue in the body. It's classified as a connective tissue, and it's divided into two categories, kind of the living cells, which are referred to as the formed elements, and then the non-living matrix, which is the plasma. So that non-living matrix is the fluid matrix of blood. And in that picture there in the middle, you see a test tube that's been centrifuged, or an illustration of a test tube that's been centrifuged. And when you spin blood, the heavy stuff, or when you spin anything for that matter, but in this case blood, when you spin the blood, the heavy stuff goes to the bottom, it precipitates. And so the formed elements fall to the bottom of the tube, which allows for the separation of the formed elements from the plasma. So you see at the bottom of the formed elements there, you have your red blood cells, or the majority of the formed elements, because they're the most abundant of them. And then that little kind of white line is referred to as the buffy coat. And the buffy coat consists of the white blood cells predominantly, and also the platelets. Platelets are just fragments of cells. And then, of course, the plasma sitting on top. So as we've been discussing, the average volume of blood is about 5 liters, which is the equivalent to the cardiac output measurement, which we pump our entire blood volume every minute. The arterial blood leaves the heart and it's oxygenated, so it has a bright red color. It's not oxygenated when it goes to the lungs, but that's a little bit of a different story. We'll talk about that when we do the respiratory system. But everywhere else, arterial blood is going to be oxygenated. It's going to be bright red. And that blood in the arteries is going to ultimately travel to a capillary where we can exchange our nutrients. The venous blood, once the nutrients are exchanged and the wastes are picked up, the venous blood is going to be largely deoxygenated, with the exception of the blood coming from the lungs again. And ultimately that venous blood is returned to the heart. If you remember, the term for that was venous return, and that blood enters the right side of the heart. So as we saw on the previous slide, about 55% of our blood is plasma, and 45% by volume, and 45% are formed elements. So in the case of the plasma, again, this is the fluid part of the blood, and you can see in that, you certainly don't need to memorize this, but there are a few important things we'll want to remember about the plasma. So again, 55% of our blood by volume is plasma, and a lot of that's water. As we know, water is our main solvent in the body. Then we have our electrolytes, and those are things like sodium, which is our primary extracellular ion, necessary to maintain osmotic balance in our body. We have potassium, which will ultimately play a role in our ability to manage our pH. We have calcium, which magnesium, chloride, and bicarbonate, all of which are important for many things, but we think about, membrane permeability, also all kinds of like muscle functions and all sorts of stuff. And bicarbonate also helps with the pH. So there's all sorts of roles that these electrolytes, or the salts that we find in our blood, play. Then we have a fair amount of protein, in our plasma. The primary plasma protein is albumin. We'll talk a little bit more about that in a moment. Albumin plays a really important role in, again, osmotic balance. Also, proteins help to buffer our blood. We have our intracellular protein buffering systems. Fibrinogen is going to play a role in the clotting of our blood. And then we've got our globulins. And globulins do quite a few things. We've got a big category of globulins called antibodies, and those are very important for our immune system's adequate function. And then we have globulins that help to transport fats around in our body. And then, of course, we've got other, nutrients. We've got waste products. We've got, you know, uh, our respiratory gases, and there are CO2 and O2. Of course, we've got hormones transported in our blood. So at any given time, the substances being transported in our bloods are going to be, a little bit different. But that's one of the most important roles blood plays is allows for us to transport these nutrients from place to place. All right, so certainly don't memorize any of this, but I put in a couple charts here just to give you an idea of, what our normal plasma values are. We know our blood pH, the range is 7.35 to 7.45, but normal is, somewhere between 7.38 and 7.44. The osmolarity of our blood gives us an idea of the concentration. And when we talked about this earlier, we used the number 300, which I said was probably a little high, but it's just a nice round number. And you can see there that, it's most people's blood osmolarity is between 285 and 295. So there's some enzymes that we find in our blood that are nice to know about. One is creatine phosphokinase. Plays a role in kind of helping to, one of the roles that enzyme plays is it helps with the ability of our muscles to continually be replenished with ATP. So the phosphate group can get broken off of something called phosphocreatinine, and this enzyme helps that process. So I will say, sometimes if somebody has had some damage to their muscle cells, and the muscle cells have been ripped apart. I'm trying to find a nicer way to say that, but the cell membrane has been ruptured. The enzymes will spill out, and so those enzymes sometimes are markers for damage to muscles specifically. So you'll see that CPK specifically is one of the markers that tells us there's muscle damage, specifically damage to the heart. That's kind of where we look for it, but it could be any muscle. It's not specific to the heart. Okay, and then go down to the hematology values. Hematocrit and hemoglobin give us an idea of our oxygen carrying capacity, how much oxygen is what the percentage of our blood is, what percentage of our blood is hemoglobin, and then what's the actual measurement for hemoglobin, and then you see red counts and white counts down there. So these are just some important things that are things that we would look at in blood. And then, of course, there's others. You can see we could look at hormones. Look at the measurement for hormones. They're in small amounts. And you can see they're measured in nanograms per 100 mils. So our hormones are not there in high amounts. And then you can see the normal plasma values for your ions. And then some of those organic molecules that are important, like cholesterol, glucose. That would be a fasting measurement, although 115 is too high from my perspective. Anyway, that's the kind of stuff we find in blood. So let's talk about the plasma proteins for a minute. This is something I would like you to remember. So of all the solutes in the plasma, these are the most abundant, and just shy of 10% of the plasma is of the solutes in the plasma are going to be plasma proteins. These proteins are made by the liver, and there's a couple important ones I mentioned earlier, but we'll talk about them again quickly. So albumin is the primary plasma protein, and so that means it's going to be the one that's in the highest amount in the blood. The reason why albumin is really important is because it regulates osmotic pressure. It basically pulls water from our tissues into our capillaries. and helps us to maintain blood volume and pressure. And the reason why protein will draw water into the capillary is that the protein draws water specifically into the venous end of the capillary, and that's because if you remember from our molecular motion talk early on in the semester, we know that water will always move towards the higher solute concentration. So if your proteins are big, right? Proteins are big molecules. And so they're not able to just diffuse. They're out of the cell, out of the blood, and into the tissues, right? They'd have to escape through some kind of a hole in the capillary, which is not something that's normally there. So if the protein is if the proteins are in higher concentration in the blood, then water will move towards that higher solute concentration. And this is, kind of the basis of the capillary dynamics, which we are discussing in our class this semester. So, it's an important thing to note that the presence of albumin specifically is kind of the force that draws water into our blood and maintains our blood pressure. Clotting proteins are going to help to prevent blood loss when a blood vessel ruptures. So they're going to be activated when there's some injury. They're not doing that all of the time. Antibodies, again, I mentioned just previously that antibodies are made by our immune system. They're made by a specific type of cell called a plasma cell, and they are made in response to some kind of pathogen. And their job essentially is to bind up, and the term for the thing that they're made in response to is called an antigen. So antibodies are made in response to antigens, and the antigen is kind of the foreign thing that's not supposed to be there, the invader, if you will. And the antibody and antigen, well, the antibody, once it's formed, has binding sites on it for the antigen, and it binds up the antigen and it makes it less problematic in the body. And then the immune system has to deal with that antigen-antibody complex, and it does that in various ways depending on the antibody and the antigen. And then we also have proteins called globulins. There's alpha and beta globulins, and their job is to transport things around, most notably lipids and fat-soluble vitamins. Our fat-soluble vitamins are vitamin A, D, E, and K, and those guys are transported, bound to these globulins. There are also things called gamma globulins, which are kind of preformed antibodies that can be functional in the immune system as well. So you may have heard of like a gamma globulin injection. If, for example, let's say you're working in a hospital and you get a needle stick, which is not a good situation. And oftentimes, depending on the scenario, so let's say you got a needle stick and the needle, potentially had come in contact with another person's body fluids that had a disease that you don't want. Like, for example, I don't know, the classic one is HIV, of course. So, or let's go with like Hep C or something like that. Anyway, so when your body is invaded by an antigen, something in this case virulent, potentially, normally what happens is your immune system gets activated to that, and you make antibodies, and your body deals with that antigen. That's the normal way it happens. That's called natural immunity. But in this kind of situation, that takes time, right? It takes some time for all that to happen. And in this kind of situation, if you potentially have been administered an antigen inadvertently, we don't want to wait and risk all of that time. So sometimes what they'll do is they'll give preformed antibody in the form of gamma globulins. And that means the antibodies that you don't have to go through the trouble of making them. They're already preformed and they're injected, and then they can look for that antigen. So it decreases the risk significantly. And then finally, fibrinogen is a plasma protein that's going to help in the clotting process after it becomes fibrin. So we'll talk about that in a little bit. All right, so the plasma volume is maintained by all sorts of regulatory mechanisms. And the reason for that is because the volume of our plasma determines our blood pressure, and blood pressure needs to be homeostatically controlled. And there's a whole bunch of regulatory mechanisms that facilitate that process. One of the ways that our body monitors the concentration of our blood is by the presence of these receptors called osmoreceptors. Osmoreceptors are sensitive to the osmolarity of our blood, or the concentration of our blood, and these osmoreceptors are found in the hypothalamus. And if the blood is too concentrated, then that will cause the release of a hormone called antidiuretic hormone. And antidiuretic hormone comes from, does anybody remember where? Hopefully you're thinking. Antidiuretic hormone, ADH, comes from the posterior pituitary. Antidiuretic hormone and oxytocin, if you recall. And so antidiuretic hormone's job is to basically cause us to retain fluid. So if the blood is too concentrated, that means fluid has been lost, or for whatever reason, there's not as much fluid there. So the role of antidiuretic, so the osmoreceptors sense that. They are in the brain. They're actually in the hypothalamus, which, if you recall, the posterior pituitary and the hypothalamus are connected. The hormone is actually made in the hypothalamus, but it's stored in the posterior pituitary. And that hormone then is released from the posterior pituitary, and it ends up acting on the kidney tubules, which we'll talk more about that in the coming weeks. And it causes water channels to be inserted in the collecting duct primarily, and that allows for water to move from the kidney tubule to be reabsorbed and put back into our blood, and it causes our blood to be less concentrated and more dilute. All right, so we know that our blood pH is probably the tightest to most tightly regulated variable we have. If the blood is too acidic, we call that acidosis. If the blood is too alkaline, we call that alkalosis. When the pH of the blood swings one way or the other, there's all sorts of things put in place to fix that too, or at least to compensate for it to some degree. Of course, we have blood buffering systems, but once the blood swings, the buffers are no longer functional, or are no longer able to maintain the pH because they're more or less saturated. And so then we rely on either the respiratory system, usually first, which you've talked about in the lab a few times, and also the kidneys second, but the kidneys are going to do a better job at helping to restore the pH, or at least bring it closer to where you want it, closer to range. All right, so I want to talk to you a little bit about these formed elements. These are our red blood cells, our white blood cells, and our platelets. So you can see on the picture there, the most abundant cell in there are the red blood cells, and they're the little red ones. They look kind of like sphere discs with sort of a pale spot in the middle, and that's because of the shape of the red blood cell. And then on this particular picture, we see 4 white blood cells, 3 of which are neutrophils, and 1 of which is a lymphocyte. And then we see those little purple specks, which are platelets. So erythrocytes, or red blood cells, are pretty small, are very small, but they're really abundant. So they're flattened biconcave discs. So that's why they're sort of pale in the middle. So they're like a disc that's kind of got like a membrane in the middle. The job of the red blood cell, as you guys know, is to carry oxygen. It's a weird cell because it doesn't have a nucleus and it doesn't have mitochondria. But there's a ton of them. You can see that, there's a lot of red blood cells. If you look at that measurement, approximately 5 million red blood cells in a millimeter cubed of blood. That's life mind-blowing, right? A millimeter cubed is a millimeter, if you were to make a little box with millimeters, which is the little lines in between, those little teeny lines on the metric ruler, you'd have 5 million red blood cells in there. These things are tiny. Because they lack a nucleus, they cannot reproduce and divide. And so they have a set lifespan, which on average is about 120 days. So in these, so this is another mind-blowing number, I think. So we've got 5 million of these things in a millimeter cubed of blood. Each red blood cell, just one, contains about 280 million hemoglobin molecules. That's in just one. I can't do the math right now because it's too early in the morning, but just imagine 280 million hemoglobin molecules in one red blood cell and 5 million of them in a millimeter cubed of blood. That tells you how excellent we are and adapted we are to carrying oxygen, right? It gives you an idea of how much oxygen we are transporting around. I'm pretty sure on the next slide we'll look at hemoglobin in a little bit more detail, but the iron heme component of the red blood cell is recycled by the liver and the spleen, and it's carried by transferrin in the blood to the red bone marrow. So it gets recycled by the liver and the spleen, and then it's carried back to the red blood cell by transferrin. So sometimes if you have like a kind of a comprehensive iron panel, if you're an anemic person and they are looking to kind of figure it out, they look at more things than just hemoglobin, right? They look at things like transferrin to make sure that you're able to actually transfer your basically recycled iron heme back to the red bone marrow so it can be, utilized again. So iron, here we go. You see it up there. There's the heme component of hemoglobin. So the iron heme is right there, smack in the middle, and that's the one that's recycled from the liver and the spleen, and it's carried by transferrin to the red bone marrow. Hemoglobin, in the middle of that picture up at the top, is an iron-containing protein. The iron, I guess, is just what we just looked at, which has got 4 oxygen binding sites. Hemoglobin is an interesting molecule, and its relationship to oxygen is pretty interesting. It binds oxygen readily. It has a high affinity for oxygen, but also reversibly. So it's like, that's not a traditional kind of binding arrangement. If something has a high affinity for another to bind, if something has a high affinity to something that it's going to bind, it tends to not want to let go of it. But hemoglobin's an interesting one. It has a high binding affinity for oxygen, but then it will also let it go, which obviously is helpful because it wouldn't do us any bit of good if we bound up all the oxygen to our hemoglobin and then it wouldn't let it go. So each of our red blood cells has a lot of, somewhere between 250 and 280 million hemoglobin molecules. A normal blood sample will have somewhere between 12 and 18 grams of hemoglobin per 100 millimeters of blood. So it's quite a bit of hemoglobin. So we care about abnormalities in our red blood cells because our red blood cells specifically, are super important to us because they are our oxygen transporters, among other things, but that's the main role. So if you have a condition called anemia, and there's lots of different versions of anemia, but you have a decrease in your oxygen carrying capacity, essentially. You can have iron deficiency anemia. You can have B vitamin deficiency anemia, which causes something called macrocytic anemia. You can have sickle cell anemia, which is a result of abnormally shaped hemoglobin. You can have something called microcytic anemia when your red blood cells are too small, so they don't have as much hemoglobin in them. There's all sorts of options. But in any of those, it compromises your oxygen carrying capacity. You can have the flip side of anemia, which is called polycythemia, which is an excess amount of red blood cells. And you can have a genetic version of that where you always have too many red blood cells, or you can have some condition that makes you all of a sudden increase your number of red blood cells. That would be potentially like if you have my brain is not awake yet. Oh, a condition called emphysema. Emphysema is a obstructive pulmonary disease, but because of what happens in the lungs and because of the fact that we don't oxygenate very well, our bone marrow will start spitting out more red blood cells. The problem with too many red blood cells is you think like, wow, that would be great because you have a greater oxygen carrying capacity, which is true, but your blood becomes thick. And if you recall from the blood flow conversation that you had in the lab, thick blood, viscous blood, this is an increase in viscosity, increases resistance, and it makes the blood flow not as efficiently and makes the heart work harder. So that's a negative. Blood doping, which we have heard a lot of over recent years, is when we basically inject something or administer some kind of a medication that causes an increase in red blood cells. So it's an artificially induced polycythemia. And the reason why this is done, again, is to increase your oxygen carrying capacity. The more oxygen you can deliver to your muscles, the more efficient your muscles, right? Muscle contraction, as we know, is really expensive metabolically. And so what happens when you are working your muscles very hard, we tend to work hard to the point of failure where you start to not have as much oxygen around. So if you can give yourself more red blood cells, you can also, sustain muscle contractions longer. Ride your bike all over France or whatever. That's the only thing that comes to mind, the dopers, but I know there's lots of other examples, but that one's probably the best, most well-known. You might be thinking, well, is their blood thick? And the answer to that is, yeah, their blood is thicker, but in this case, these are highly conditioned athletes. So I guess I don't know. I would like to see any data on whether if the heart's actually hypertrophied, if they actually got bigger in these people, but obviously they're still using their hearts, so we can't have them. But anyway, I don't know the answer to that question, but it's one that I wonder about quite often when I talk about this. All right, here's a couple pictures. On the left of the slide, that's normal blood, normal hemoglobin, and on the right, when we don't have as many red blood cells, that would be an example of anemia, or if your red blood cells that you do have don't have as much hemoglobin, that would be another example of anemia. Here's a picture of what a sickle cell looks like. So when it sickles, the one thing that's wrong is the hemoglobin is abnormal, but the other problem is the cells are shaped weird. And because they're shaped weird, they tend to, one, stick together more often, and that increases their risk of blood clotting. And they also tend to get stuck in our tissues, our lymphoid tissues, most notably the spleen. And so people with sickle cell anemia oftentimes have splenic enlargement, and their spleens can rupture as a result of that. Aplastic anemia is a type of real serious anemia that oftentimes is a consequence of either drug therapy, but most notably chemotherapy, because chemotherapeutic agents target rapidly dividing cells, and our bone marrow is one of those locations where we have rapidly dividing cells. And so with aplastic anemia, you see definitely a decrease in red blood cells and many times platelets as well. All right, so here are our white blood cells. We've got the neutrophils, the eosinophils, and the basophils on top. Those are referred to as granulocytes because they've got those little speckles inside the cell, in the cell cytoplasm. And then we have the lymphocytes and the monocytes. Those are called agranulocytes. And then we have cell fragments, which are platelets, and then we have the red blood cells, which are not leukocytes, but I couldn't find, this picture had them on all together, so left them in there. So the role of the white blood cell, there's a myriad roles of white blood cells because they all kind of have a different job, but just as a general rule, we know that our white blood cells are very important for our ability to defend ourselves against diseases. These are complete cells with the nuclei and organelles, so they're not like red blood cells in that. Remember, red blood cells have no nucleus and they have no mitochondria. These cells are very interesting in that they have the ability to move out of blood vessels. So they travel in a blood vessel to an area of interest or damage, and then they're able to kind of squeeze out and move into the surrounding tissue. And the word for that type of transition from inside of the blood cell to outside of the blood, or pardon me, inside of the blood vessel to outside of the blood vessel is called diapedesis. And they kind of move like these big blobby amoeba things. It's pretty cool. You could go onto YouTube if you wanted and type in white blood cell migration or diapedesis, and you can see these like big blobby cells kind of squeezing out between platelets, between endothelial cells in a capillary. White blood cells respond to chemical signals. And so when a tissue is damaged, it releases a variety of chemical signals. And those chemical signals are sort of the call to action for these white blood cells to come to that area. So the migration of a white blood cell to an area of damage is called chemotaxis. So we don't have nearly as many white blood cells as we have red blood cells. Remember, we have 5 million per millimeter cubed for red blood cells. For white blood cells, we have somewhere in the neighborhood of 4,000 to 11,000, kind of depending on the status of the person's immune system. So just for some general abnormalities in terms of numbers of white blood cells, again, this doesn't give you a whole lot of information because each white blood cell kind of has its own job. But if somebody had, if you were looking at someone's blood and you were just looking at a white count, which would be all of the white blood cells found in that sample of blood, a white blood count over 11,000 leukocytes or white blood cells per millimeter cubed is referred to as leukocytosis. That just means lots of white blood cells. And that generally means they have some kind of an infection. You can't really tell what sort of infection. Is it bacterial? Is it viral? Is it fungal? You don't know by just the absolute count of white blood cells. An absence, a low level of white blood cells or deficiency would be called leukopenia. And that oftentimes is caused by suppression of the bone marrow by certain drugs. Corticosteroids do it, and a lot of people take corticosteroids for a variety of reasons, and a lot of the chemotherapeutic and anti-cancer agents will do this as well because, again, that's a tissue that's targeted. Leukemia is a actual type of cancer that is a cancer of the bone marrow. And what happens, and there's lots of different types of leukemia. There's acute types and chronic types and all sorts of different varieties. But what happens is traditionally the person's bone marrow starts producing an abnormal number of specific kinds of white blood cells. So that you again, here what you would probably see first if somebody had leukemia and all you were looking at what is at the white count, you would have elevated white counts, and then you'd have to go in and this is kind of just what you'd need to do regardless. So anytime you see an elevated white count, the more pertinent question is which cells are actually elevated, and that's where you start to be able to like distinguish between potentially a bacterial infection, a viral infection, and then something like leukemia by the types of cells that you find there. All right, so in that box, you see up at the top in the middle a cell called a hemocytoblast. That's a stem cell. So basically that's the stem cell that all blood cells differentiate from. So we've got these hemocytoblasts in our bone marrow and hanging out there waiting to decide what they're going to ultimately become. So the options are, in terms of white cells, are the granulocytes and the agranulocytes. And like I said earlier, the granulocytes on this picture are on the far right. They're the ones with speckles in their cytoplasm. They are the basophils, the eosinophils, and the neutrophils. All granulocytes have those granules that oftentimes are stained, so you see them under a microscope, and they stain different colors traditionally. We'll talk a little about that in a minute. They also have a multi-lobe nucleus. So their nucleus, so what you see in those cells there, it's hard to see in the basophil because it's dark, but if you see that eosinophil, that thing that kind of looks like a, I don't know, balloon animal, same with the neutrophil, that's the nucleus, but it's got multi-lobes. And there's some evidence, especially with the neutrophils, that the more lobular the nucleus, the older the cell, the longer it's been around. The agranulocytes, which are the monocytes and the lymphocytes at the bottom layer on the left, those ones lack granules in their cytoplasm, and their nuclei is usually round, especially in the case of the lymphocyte, and it tends to be big, almost taking up the entire cell. They can be oval or they can be kidney shaped, like the one you see in the monocyte there. And then while we're on this slide, oh yeah, those are the granulocytes and those are the agranulocytes. So you notice that hemocytoblast at the top. So all blood cells are going to differentiate ultimately from that cell. And then you see from there, we differentiate into either a myeloid stem cell or a lymphoid stem cell. Everything with the exception, all blood cells and platelets, with the exception of the lymphocyte, ultimately differentiate from the myeloid stem cell. So myeloid stem cells are mostly differentiating into erythrocytes, but they can also differentiate into these other cells as well. And then finally, and remember, this is all going to be driven by chemical signaling. And then the lymphoid stem cell differentiates into only lymphocytes. That's it. All right, so I want to talk about just not a whole bunch, but a little bit of each one of a little bit of detail about each one of these cells, the most important things that you kind of want to remember. So the neutrophils are granulocytes, and they've got a multi-lobe nucleus, and they have all these little granules in there that are sometimes like blue or purplish. It's hard to really know the color exactly. These guys are most abundant in a normal blood sample. So these are the ones that, of the white blood cells that are running around, most of them are going to be neutrophils. They're phagocytes. They, at a site of infection, they show up and they just eat up whatever's not supposed to be there. They're not particularly picky. They just eat it up. Eosinophils, these are the ones that the granules are brick red, and eosinophils are not found at all in a normal blood sample. I mean, you might find 0 to 1. Eosinophils, if they are present, generally indicate that either one, someone is allergic, so somebody who has allergies, and if they don't have allergies and they have eosinophils, you got to think about parasites. Parasites are parasitic infection or whatever occupation is the word I'm looking for. I guess if you have parasites inhabiting you, your eosinophils will elevate. So, and parasites are more common than we like to think, unfortunately. So pay attention to those eosinophil cells. They shouldn't be there. Basophils also shouldn't be there. Basophils have a lot of inflammatory mediators in their granules inside of their cells, but most notably histamine, and histamine's a big player in initiating an inflammatory reaction. So people who have a lot of basophils around also tend to be allergic. They have like these histamine reactions. Basophils are the ones that can leave, remember all these cells can leave all these white cells can leave the blood vessel and move into the tissues. And when basophils move into the tissues, we call them mast cells, M-A-S-T. Lymphocytes, these guys are the ones with a big nucleus, and these guys are really important in our specific immune response. They are things that are not phagocytic. They don't just eat up whatever they find. They only go after specific things, and they have to be told that. And a whole arm of the immune system activates these cells and and kind of initiates them to to a particular antigen. So they are very specific. They're like your specific army, right? They go and they find someone. They're looking for one thing, and then when they find it, they deal with it, and they deal with it a little bit differently. The B cells are the ones that make antibodies, and the T cells are able to like kind of engage in like sort of what would be the equivalent of cellular hand-to-hand combat, where they like, it's like cellular warfare, where they'll like spit out like things that cause cell membranes to rupture. It's pretty cool. And then finally, the monocytes are the biggest of our white blood cells. They are also phagocytic in the tissues. So monocytes leave our tissues and they become what are called macrophages, which I'm sure you've heard of before. So a monocyte in the a white blood cell in the a monocyte in the blood vessel becomes a macrophage in the tissue. And again, these guys are helpful for fighting chronic infection because they kind of clean up, the byproducts of a chronically infected tissue. But they're phagocytes. They're not particularly picky either. All right, platelets are fragments of the big cells called megakaryocytes. Because they're cell fragments, they don't have a nucleus. They don't stick around long. They only live like a week or so, give or take a couple days. They do play a role in blood clotting with a couple other chemicals. The platelets make the plug. And then the plug gets sort of held together with fibrinogen. Platelets release serotonin, and when a tissue has been damaged, platelets can release serotonin, and that stimulates the vessel to constrict, which minimizes any further blood loss out of that tube. And you can see the count there, somewhere in the neighborhood of 130,000 and 400,000 per millimeter cubed of blood. All right, so again, this process of making blood cells from that hemocytoblast is called hematopoiesis. So hemato means blood and poiesis means to make. So the process of making blood occurs in our red bone marrow. And again, all of the blood cells are derived from that common stem cell, and that's common stem cell is called a hemocytoblast. It's that one at the top again. And then you can see it will differentiate into the myeloid stem cells and the lymphoid stem cells. The lymphoid stem cells are going to produce lymphocytes, and the myeloid stem cells are going to produce all the other blood cells. And they continue, so they start as these cells, and then they continue to differentiate. And as they continue to differentiate, they will ultimately develop membrane receptors for all the various chemical signals that will ultimately call them to action. It's a pretty complex process. The red bone marrow is going to produce these red blood cells, and it's going to produce red blood cells at a rate of about 2.5 million red blood cells per second. And they don't have nucleus, so they do not divide, they do not grow, they do not make protein, they don't do any of that stuff. They wear out in about 3 months, 100 to 120 days, and once they're done, they get recycled by either the liver or the spleen. The important parts get kind of taken out, and then those important parts ultimately get recycled back, usually to the red bone marrow, so you can use it again. And we're constantly, so if we're making like all per second 2.5 million red blood cells per second, we're also losing them about the same rate, right? Because your count stays steady. So as we lose red blood cells, we make more. So again, it's that hemocytoblast that's constantly differentiating in the red bone marrow to replace all these red blood cells that are lost. This process of erythropoiesis is controlled by a hormone that's actually produced in the kidney called erythropoietin. So erythropoietin is produced by the kidney in response traditionally to oxygen levels in the blood. So your blood is being monitored for oxygen concentration, and if the oxygen concentration is low, then erythropoietin is produced by the kidneys and it travels in the kidneys um, until it ultimately basically triggers this process, which is to um, stimulate the differentiation of the hemocytoblast into ultimately erythrocytes. And so you can see the kind of process there is starts at the top of the hemocytoblast, and then it goes, that differentiates into a myeloid stem cell, which then differentiates into a pro-erythroblast, and then an erythroblast, and then what we call a normoblast. And then at that point, the nucleus gets spit out, and then we have a nuclear cell called a reticulocyte. A reticulocyte is an immature red blood cell, and it is the last step above the erythrocyte. And the reason why it's kind of a good one to know is because if for some reason a person is needing to produce a lot of red blood cells faster than they're really prepared to do, what will happen is ultimately we'll start seeing reticulocytes less released from the bone marrow before they're ready. So they're just basically releasing things so quickly that the reticulocytes get released into circulation. And so when you start to see reticulocytes in blood, it gives you an idea that you've got all these immature red blood cells around, which leads you to then think, why would we have immature red blood cells? What's going on? And so your first thought is either they're losing blood or their bone marrow is being stimulated too much. So if someone's losing blood faster than they can make it, they're taking blood cells out of circulation faster than they can make them, then you get these immature cells released. Or if something's stimulating their bone marrow to divide to do this process too quickly, then sometimes we'll see reticulocytes in blood, in bone marrow, in the blood as well. So the presence of reticulocytes always, should raise a level of concern that there's something going on there that's outside of, normal homeostasis. Here's a little kind of a graphic that gives you an idea. So we've got our little homeostatic, teeter-totter there, and when oxygen levels are normal, we're balanced, but when they fall, if the oxygen levels in the blood falls, or if there's a decrease in the red blood cell count, or if there's an increase in the demand for oxygen by your tissues, all of that can cause this to cause this to tip. So it all kind of equates to reduced oxygen levels in the blood, and that's what the kidneys are sensitive to. So the kidneys release erythropoietin, which stimulates the red bone marrow to produce more red blood cells. And then those red blood cells are released into the blood, our oxygen carrying capacity increases, and we sort of balance out there. All right, I just want to finish, and I want to talk about clotting for a bit. The term for blood clotting is called hemostasis, and the purpose of a blood clot is to stop bleeding from a ruptured vessel, of course. Blood clots usually form pretty quickly when a vessel has been damaged, within a few minutes, and the blood clot will remain there until that inner cell lining of the vessel repairs. The inner lining of the vessel, as you remember from anatomy, is called the endothelium. So the clot remains in location at that place where the damage occurred until the endothelium regenerates. And then the clot's broken down after the tissue has been repaired. So without getting, there's lots of little steps in the formation of a blood clot, which we're not going to really dive into, but just generally, there's like three main phases of blood clot formation. One is the spasm of the vessel that's been damaged. Two is the formation of a platelet plug to kind of plug the hole. And three is the coagulation, the blood clotting to make it so, it sort of like cements the hole up. So you see in this picture here, you've got an injured vessel. The injured vessel releases a chemical signal called tissue factor. Tissue factor interacts with a phospholipid called PF3, and when tissue factor and PF3 interact with one another, clotting factor, along with clotting factors and calcium, triggers a clotting cascade. And now, again, this is where it can get, that's where really all the details are, is in that whole blood clotting cascade. So we've got up there, you see your blood injured vessels. You see then another chemical signal called prothrombin activator. The role of prothrombin activator is to convert prothrombin to thrombin. But in order to do that, you need calcium. That's one of the roles of calcium. So calcium is necessary for the conversion of prothrombin to thrombin. Thrombin, once it's produced, provided there's calcium around, thrombin then joins with proteins called fibrinogen, and it makes these hair-like, kind of like tough strands of protein, insoluble protein called fibrin. And then fibrin's job is to kind of make a mesh, like a meshwork that will allow the platelets and the blood clot to sort of stick to that area. So fibrin is the mesh that sort of holds it all there, and it's going to stay there. It will stay there until the endothelium regenerates underneath. So here's another picture. So we see the damaged vessel releasing clotting factors, and clotting factors are also dependent on calcium. That's where all that is. The clotting cascade is got, like, I can't remember off the top of my head how many steps, but there's a ton of them. And there's other things, like you need vitamin K, you need calcium, you need all sorts of things for this to happen. So the clotting factors and calcium primarily are needed for the conversion of prothrombin to thrombin. Thrombin then's role is to convert fibrinogen, which is soluble, floating around in the plasma, into fibrin, which is the insoluble kind of, so the thrombin and the fibrin, join together, and they make this insoluble sort of like tough, kind of like thread, but tough. And these fibrin strands stick to the platelet plug. So there's platelets there, they're all stuck together, there's red blood cells stuck to the platelets, and then we put the fibrin, which sticks it all together, and it's plugging that hole where the damage occurred. I didn't mention, but you see it in the middle picture there, that the vasoconstriction, you see that remember, it was first vascular spasm. It was one of the first things that happens to kind of minimize any more blood leaking out of there. So then those fibrin strands adhere to that. Now we've got this insoluble clot, and that clot will stay there until the endothelium regenerates, which can be a long time depending on the size of the damage. So here you see a picture of, this is within a scanning electron microscope, and you can see the red blood cells kind of trapped, and their platelets are what kind of sticks this whole thing together. And the fibrin is, traps it all in there, right? So one of the things that sets this whole ball rolling is that when endothelium is damaged, so when a blood vessel is damaged, it exposes the collagen beneath, and that exposure of the collagen is kind of what sets off all this cellular, this chemical cascade that calls the platelets and gets the whole ball rolling. Platelets bind to the collagen, and they're held there by something called von Willebrand factor. Without going too much in the weeds, but von Willebrand factor. So there's all sorts of blood clotting disorders. Probably the most famous one that people have heard of is hemophilia. There's different versions of hemophilia, A and B, and hemophilia is one of the A is one of the sex-linked disorders. So you'll see it run in families, and there's some interesting, if you're a history person, you probably learned the history of the Russian family of, I can't remember what the name of them right now, which Nicholas it was, but it was the Tsar and a lot of the family, the you know males were not making it, and it was because they had hemophilia in their family, and they had a tendency, which in that time royalty often did, to sort of reproduce with their own, keep all the money and all the stuff in the family, which seemed like a good idea until you learn about sex-linked disorders. So anyway, that's a different story, but you history people probably have heard about that. However, so that's a big time blood clotting disorder where people with hemophilia A really don't clot efficiently at all. But then there's all sorts of other variations of blood clotting disorders. And the most common one is something called von Willebrand. It affects von Willebrand factor. And it's so common. And it is also, what's the genetics on that? I don't believe it's sex linked. I can't remember off the top of my head. I should know that, but I've forgotten now. But it is, actually, I'm going to look it up right while we're talking. Anyway, it's not a super serious blood clotting disorder, and then oftentimes people don't even know they have it. But it can be a problem if, for example, sorry, I'm looking at it right here. It is hereditary. There's different versions of it. It is a hereditary. It doesn't say. Okay, I'm not going to keep going too much, but it does look like it's. Oh, it is. It's autosomal. Most of the types are autosomal dominant, so it's not sex linked. I was right. Yeah, so it's an autosomal dominant condition, which is highly heritable, but not necessarily sex linked. So anyway, the reason why I'm mentioning it is because it is quite common, and it just retards people's ability to clot, but not to the point where they bleed to death generally. The only time, it would be really nice to know if you're going to do like surgery on somebody, because there's going to be a potential that there might be an issue with their ability to heal or bleed in this respect. But the most common symptoms of von Willebrand's would be like things like maybe nosebleeds, like frequent nosebleeds, which lots of people have. And also in females, like very heavy menstrual bleeding is another potential indicator that somebody might have von Willebrand's. I believe like 23andMe, some of those like, direct to consumer genetic tests actually test for von Willebrand's. I'm quite certain 23andMe does. Anyway, it's just sort of interesting to know. So there's all kinds of varieties of blood clotting issues. Some are very serious, some are not as serious, but they're always good to know about. But anyway, that's a blood clot. So the damage to the collagen is exposed. The platelets bind to the collagen. They get stuck there by von Willebrand factor, unless you don't have it, and then they still will stick. They'll still, form a plug, but they won't stick as efficiently. And then more platelets, platelets sort of act in like this feed forward way. So more platelets will recruit more and more platelets to come. And so the platelets just keep coming in. And some of this is driven by the collagen and some of it's driven by chemical signaling. But ultimately you make a plug and then the red blood cells stick, and then the fibrin like lays on top of it. Anyway, that's it. That's all I got for you for blood. There's a study guide posted, and I will see you soon.